For a long time, the scientific community viewed the human brain as a biological island, a sophisticated command center fundamentally isolated from the rest of the body's systems. However, a landmark study published in the journal Gastroenterology in 2011 completely shifted this paradigm. Conducted by researchers Stephen Collins and Premisel Birchik at McMaster University, this work has become a true classic in the field of the gut-brain axis. It provided the first definitive evidence that the microscopic inhabitants of our digestive tract can determine what we might call our personality or behavioral phenotype. To demonstrate this, the researchers focused on two groups of mice with genetically opposite characters. On one hand, the BALB-TAC mice, which are naturally shy, anxious, and hesitant in new environments. On the other, the NIH Swiss mice, known for their bold, adventurous, and highly exploratory temperament. The extraordinary heart of this experiment was the fecal microbiota transplant performed on germ-free mice. By swapping their microbiomes, the scientists literally swapped their behaviors. When the timid mice were colonized with the bacteria of the bold ones, they began to explore with unprecedented courage. Conversely, the once adventurous mice became guarded and fearful almost immediately after receiving the microbial community of the anxious group. This transformation was far more than a change in mood. It was a profound shift in brain chemistry. The team noted that these behavioral changes were accompanied by significant variations in the levels of BDNF, or brain-derived neurotrophic factor, within the hippocampus. This region is a crucial center for governing anxiety and cognitive functions. It confirmed that signals originating from the gut microbiome are capable of modulating gene expression and the very biochemistry of the brain. This research paved the way for the modern study of psychobiotics, showing that the microbiome is not merely a passive guest, but a true orchestrator of behavior. It is fascinating to see how these chemical signaling pathways intersect with the communication routes between our commensal bacteria and the central nervous system. We are coming to realize that our health and our very identity are the result of a collective harmony between our human cells and our microbial partners.